Hello, everyone. Welcome to the webinar. It is my pleasure to introduce Dr. Mark Moorman, who will now give our opening address to everyone to begin the webinar. Thank you.
Well, hello, everyone. My name is Mark Moorman. I'm the director of the Office of Food Safety at the Food and Drug Administration. And it is just my pleasure to start us off today in discussing the important topic of water and water reuse. Just a little bit of background. The Centers for Disease Control in the United States estimates that 48 million people get sick in the United States, and 128,000 are hospitalized, and 3,000 die from foodborne illness each year in the United States. And as an agency, we're talking a lot more about water. We have had some significant foodborne illness outbreaks in which water may have been a source or a vector for foodborne illness due to a whole host of pathogens. We have gone through, really since 2006, issues with water, concerns with water and E. coli with leafy greens in Santa Maria, California, salmonella from onions. We had over 1,600 people in the United States and Canada get sick from salmonella within red onions. And we had a very large cyclospora outbreak due to that we linked and actually discovered cyclospora within irrigation water, canal water next to farms. So we are talking a lot about water, and I've come to terms as I've been with the agency and have had Kruti Rebelaya within our team educate me on water that, like most Americans, I grew up taking water for granted. While only 3% of the Earth's water is fresh water, I'm from the great state of Michigan. For all of you that are not familiar with it, that's what Michigan looks like. Where while only 3% of the Earth's water is fresh water, the great state of Michigan is surrounded by lakes, and it's 20% of the world's surface freshwater. I didn't know anything about water conservation or reuse simply because I didn't have to. And I'm not alone in that. I'm absolutely convinced I'm not alone. Yet I've come to realize throughout my career that we have significant reasons for us to revisit these assumptions. Climate change, we have an aging infrastructure. I'm from Michigan, as I said, where we've had tremendous issues with lead in water that was linked to the infrastructure and processes used to maintain the infrastructure. And very importantly, we have very concentrated agriculture that's in desert regions throughout the United States. This presents major headwinds for us that is forcing us to rethink our relationship with water. Now, there's very good news in this space in that other countries, quite simply, are very far along on considering water and water reuse. Israel is a global leader in the culture of water. And I do mean culture because that's really what it starts and ends with. It has a culture of water that is embraced by its people, it's technology-led, and it's supported by its leadership. And we admire that. And quite simply today, we're anxious to learn more and to share. I want to recognize the leaders at the EPA, that them with other stakeholders have pulled together this partnership with diverse groups with this focus on agriculture. This marks the 30th year of technical cooperation on environmental management between EPA and the Israeli Ministry for Environmental Protection. That is incredibly impressive. This natural water reuse action plan collaborative represents the efforts of more than 100 partner organizations seeking to build financial, technical, and institutional capacity for communities to pursue water reuse practices. As I said, we have a lot to learn, and there is some success that will be shared. The FDA looks forward to the knowledge sharing opportunities with Israel as we continue our discussions on the last day of this meeting. Israel can share its useful information in the implementation of water reuse for agricultural purposes. And I'm thrilled to see, as I've looked through the work of this group, the case studies that provide tremendous learning opportunities. One of the things our office is doing, and we're telling a story. What happened? We're trying to get people through discussing these case stories. We're trying to be much more transparent as an agency because people need, like most things, to get it before they can really get it. Surveys show that acceptance of water reuse programs are positively influenced by an increase in water reuse knowledge, higher levels of education, and the secret sauce of all this is trust in technologies and water professionals. This water reuse will continue to be important to our work as an agency and, of course, to our colleagues within EPA. And with future potential water shortages, growers are continually searching for water options. There's a good story in California with the Irvine Water District and the city of Santa Rosa have both used reclaimed water to irrigate lettuce, strawberries, grapes, and other row crops for more than 30 years. So it can be done, but it requires groups like this to say it can be done and to teach. And so we're just thrilled to be a part of this. I want to thank all of you for bringing your knowledge and desire to share to the table today. So with that, thank you and look forward to listening.
Thank you, Mark, for those opening words. Very nice. And indeed, I mean, I'm Adam Shalimzik. I'm head of the International Relations Division at the Ministry of Environmental Protection in Israel. And I want to say this cooperation with the EPA that we have now had for about 30 years is really a true, it's a thing that we learn so much from. And we are happy that with this important webinar, we are also sharing a bit of our experience on water issues with the US. So this event is truly a highlight of our cooperation with EPA in recent years. And it's so important for us to create these connections with all relevant stakeholders in the US. So, today we have some remarkable speakers who will present different aspects of irrigation, of agricultural irrigation with reclaimed wastewater. And I must say, since we have a relatively long list of presenters today, I want to ask all speakers to please respect the time limit, and I will be reminding you of the time. Okay, so our first part today is focusing on the implications of agricultural irrigation with recycled water. For those of you who participated in the previous 2 days, you might hear some things at the beginning that you already heard regarding regulation issues, but this is just in order to lay the basis for the advanced research that will be presented after that. So the first speaker of today is Mr. David Weinberg. David is a National Planning and Treated Effluent Reuse Manager at the Israeli Ministry of Health. David will talk about supervising treated effluent reuse. David, the floor is yours, please.
Hi, good morning. First of all, I would like to say thank you to the organizers and everybody that has joined us for expressing interest in the water and effluent solution in Israel. If you could please flip. I beg the pardon of who heard me in the first day because I won't be deviating too much. Please flip one more. One more flip. Yeah, thank you. A bit of background. Israel is small. It is only 5% of California in size. We have 9 million inhabitants and that live mainly in the center of and the north of Israel. So it is densely populated. The climate is semi-arid and the south is desert, similar to New Mexico. And in the center and north, we have about 500 milliliters of rain per year and even more. But the rain falls only in the 3 to 4 months of winter time and during a total of only a few days. So much of the rain falls and reaches the sea. Please flip. Okay, so in the 90s, we had a shortage crisis, shortage of water, and it was solved quite efficiently by building desalination plants and by effluent reuse in agriculture. Our drinking water is supplied from wells, the pink dots you see, and from seawater desalination plants. One must say that Israel has a structure, a long structure, and we are beside the Mediterranean Sea, so this is possible. We are very proud of the quality of drinking tap water in Israel, and the Ministry of Health did not approve of potable effluent reuse because we preferred a safer water source, and this solution is better suitable for Israel, using regulatory and natural barriers to assure public health. If you could please flip. Okay, so we have 370 MGDs of sewage produced and are treated in 90 big wastewater treatment plants that treat over 90% of the sewage and about 300 small wastewater treatment plants. 85% of the effluent is reused, mainly in agriculture and some in public gardens and in industry. And 50% of the effluent is tertiary quality. Today, 45% of the water in agriculture is treated effluent. If you could please flip. This reuse requires monitoring in 3 levels. The first one is monitoring industrial sewage that goes into sanitary public sewage. There are 37 chemicals that are monitored, and the factories are fined for deviations from the required levels. If you could please flip. The second level of monitoring is the monitoring of sewage and treated effluent in the wastewater treatment plants. One before, please. Flip one before, please. Our 2010, no, flip backwards. Okay, yes, backwards. Backwards, please. Yes, that one, please. So our 2010 quality regulations, we shortly call the 10-10-10-10 regulations. That is because 10 milligrams per liter of BOD, of TSS and ammonia nitrogen, and 10 fecal coliforms per 100 ml. And this is about 50 times less stringent compared to California regulations. In most wastewater treatment plants, the technologies activated sludge plus deep sand filtration and chlorination with a minimum contact time times concentration, the CT of 30. And this is 15 times less stringent than the California regulation, but it is enough to reach the 10 fecal coliform and per 100 mLs and 6 log of virus reduction. This quality is similar to the quality demanded by the new 2020 European regulations for effluent that can be used to irrigate raw eaten vegetables as allowed in Israel. So BOD, COD, TSS, and fecal coliforms are sampled twice a week by a Ministry of Health approved private laboratory that reports online to the Ministry of Health, to the Ministry of Environment, and to the Water Authority. Please flip. The wastewater treatment plants need to report online monitoring parameters such as turbidity and chlorine residual via an internet application. And the reporting computer software has a BI function that helps the Ministry of Health, Environment and Water Authority to stay informed of treatment levels at the wastewater treatment plants. Could you flip, please? One more. And one last one. Okay, that's the one. The third monitoring level is on the effluent users. Using treated effluent requires a permit. It is illegal to use effluent without an annual permit. Effluent irrigation started to grow rapidly in the 80s, and today we at the Ministry of Health give over 4,000 permits per year. Each plot borders are outlined in the GIS, along with the data regarding the effluent source and the crop growing in the field. The map you see on the left, you can see the purple areas are the secondary and tertiary treated effluent, and the brown is the even higher treated effluent from soil aquifer treatment of the Shafdan wastewater treatment plant that we talked about yesterday and the day before. Please flip.
David, you have 3 more minutes.
Okay. This needs a good crew of inspectors that meet the farmers and help them fill in the forms and map and make environmental sampling. Flip, please. The farmers can choose from what to grow in taking account the treated effluent, the type of irrigation, they have to, this is influences the distance between the effluent and the produce. Please flip one more. And the combinations, the different combinations the farmer can choose from are taken into account in our 1999 application regulations that take all the characteristics such as is the vegetable eaten cooked and does it have a non-edible peel. So the inspector makes sure that the distance between the irrigation and the public is kept. He must check for possibility of cross connections. He looks into do they have alert signs, and does the farmer use purple pipes? Please flip. Monitoring is also made in reservoirs, and we take in account the complex piping system that moves effluent between the reservoirs according to where it is needed, and monitoring is also in public gardens. Please flip. Some effluent is used in industry, mainly in wetting dirt roads in quarries and in power in cooling towers in power plants. Please flip. The Ministry of Health also monitors polio routinely to prevent outbreaks, and at the last year we were monitoring corona RNA to help prevent the outbreaks, and we monitor also RNA sequencing to collect data on what different variants are there. The corona is quite low now because of vaccination, but we hope this will help keeping on checking it will help monitor another next outbreak. Okay, please flip. At the last few years, there was lots of research about micropollutants, especially the CECs, chemicals of emerging concern, including pharmaceuticals. There was much research about that, and we made, I'll be happy to talk about this later on in the discussion, but we made a health risk assessment regarding carbamazepine, but there's a lot more to be checked in and to be researched in this field. For now, we don't monitor this, not here, just like in the United States and in Europe, we don't monitor this. Please flip. And one more, please. Just tell about what we talked about yesterday, a new plant, a new model we are planning in the Shafdan for 32 MGDs, which will have membrane, bioreactor, ozone, biological activated carbon, and UV, and we'll have to reduce 8 logs of crypto and giardia and 10 logs of viruses, and reduce 80% of CECs. And this is what we are checking now in a pilot plant that was built. So thank you very much.
Thank you very much, David. We now turn to Professor Uri Yirmiyahu, who is Deputy Director for Research and Development at the Volcani Institute, which is the agricultural research organization of the Israeli Ministry of Agriculture. Professor Yirmiyahu will present principles for application of reused water in agricultural irrigation. Uri, you have the floor, please.
Thank you for the organizers, and hello everyone. Flip please. Next one. I would like to start with the terms, and you can see here a list of terms of reclaimed wastewater, treated wastewater, and so on. And we are using the word effluent, but each of these terms can be used. Maybe effluent is the shorter one. Next, please. And okay, one thing we must remember when we are dealing with water, and mostly with effluent, is 99%, maybe more, is H2O. This is water. What we are worried about is what we have more than that. And you have here all the list of things that we can find in the effluent, and it's starting from microorganisms, pathogens, and so on. And this is something you saw in the last 2 days. Next, please. But all the red one, this is something that we would like to avoid. We don't want to have these materials in our water, irrigated water, or any water that we are going to use. And it's bunch of things, and of course, we need to check all of our water, and sometimes during the time, because one analysis for a year, it's not enough. And this is something that just when you know what you have, you need to deal with the problem. So you're not going to deal with the problem that if you don't have sodium chloride. But if you have sodium chloride, you need to know that you have sodium chloride and deal with this problem. The yellow things is nutrients and trace elements. We already have them, and this is advantage. And we need to use this advantage about minerals when we are talking about irrigation. Next, please. Another thing that in Israel, our effluent characterization is a fact from 2 main things, is the wastewater content, what we start with, and the treatment type. And David just explained that we have different treatments and it's changed during the time. So again, in order to know what you have, you need to know the basic, you need to measure all the time and think what you need to do in order to irrigated effluent water. Next, please. So why in Israel we are using irrigation? Just the next. David just mentioned numbers, and here you can see the trends. So almost 40 years ago, we started to use effluent for irrigation. At the beginning, it was small amount of water. If you compare it to the total amount that we are using in Israel for irrigation, you can see that in the last 40 years, the amount that we are using every year it's decreased. Maybe we are in the plateau, but this is the a way of improve our efficiency. At the same time, the fresh water amount that we are using, it's decreased. Today, it's something between 40 to 60%, but 50% in average of our total irrigated water is from effluent. In the last 40 years, Israel use a lot of energy and support to learn what the effluent doing in different subjects. It's starting about the soils. We have different soils. We have different crops, different management, of course, different effluents, characterization. And we have special committees that trying from the beginning to learn for short term and long term the effect of the effluent for agriculture. We started slowly using the effluent, but as you can see, from year to year, we have more water that used for irrigation, and we need to know what this water doing to our soil, to our environment, and we have today, I think that the knowledge that we have in the last 40 years, it's something that we can share, and actually everything published, so it's easy to look in the literature. Please, next. If we are looking there for the total aspect of effluent, I choose 6 different subjects. Maybe you can find another one. Of course, economy. In Israel, we are paying for each drop of water that we get from the government or from the country or from the water supply. Water, it's not for free. So the economy question was how much to pay, who is going to pay for the water treatment. It was an issue. Today we pay little bit less money for effluent comparing to fresh water. The other aspect is the health, and David talked about it, so I'm jumping to the next one. It's irrigation system operating. Again, problem that happened to our system, mostly dripper and sprinkler, and this also something we need to deal with, because if the sprinkler are blocked, so you're going to change or do some treatment. So this is also an aspect that treated during the years. I would like in my talk to concentrate it in 2 other aspects. It's the environment and agronomy. Flip it. In response to environment, so we have the soil, of course. We need to look about the chemical, physical, microorganism, and you are going to hear about it, what the effluent are doing to the soil. For each part of that, sometimes chemical and physical are combined together, and microorganism, of course, and it's coming to be complicated. I can tell you in general that after 40 years of research, we find few problems, mostly in heavy soils, and I think my friends are going to talk about it. Another issue is the river and groundwater and lake. Of course, when you are using effluents, that including all these toxins, they are coming to this water, other water. And of course, the chemical microorganisms are affected by that. So this is something that all the time, I think David mentioned it, are under test, analysis. Just for example, nitrate in our groundwater and the lakes. Next, please. In response to agronomy, I found 3 main aspects of the nutrition, salinity, and toxicity, and I will start with the nutrition. Almost it's obvious that when you have water that include nitrogen and phosphorus and potassium or other elements, essential elements for plants, you can say this minerals is there and I need to take it into account when I irrigate with this water. It's take many years to, after we talk with our farmers, that they are going to, and they are taking today into account the minerals that they have in the effluents. I personally ran few experiments, field experiments for many years in order to show that I have no differences between ammonium or potassium that came from the effluent comparing the potassium that I apply as a fertilizer. So I'm very happy that today, I think most of our farmers take into account these minerals. And this is affecting, of course, the efficiency of the fertilizers. They affecting the contamination of our water, mostly nitrogen and phosphorus. But this is the practice today. I would like, next please, concentrated on toxicity. One of the main problem when we start to use effluent for irrigation was boron toxicity. We found that the boron, which is microelements, but is a tricky element, it's coming to be toxic very fast. That the concentration in the irrigation water, the effluents, are high, sometimes higher than 1 ppm. Just the level that we are using today, it's maximum 0.4 ppm. And we start to be worried that using this water is going to be first toxic to the plants. This level is not toxic to human, but more problematical, it's the accumulation of the boron in the soil. And to leach this boron up to the soil, it's difficult. So from the beginning, we tried to find a solution for boron, and we found that the high concentration of boron coming from mostly from detergent that we are using in our life. And I think 30 years ago, maybe 25 years ago, we changed the regulation, and today all the detergent are not including boron. They changed it to phosphorus, and we have high phosphorus in our effluent, but at least the boron toxicity problem was solved. Next, please.
Uri, sorry, you have 5 minutes more.
Oh, thank you. So in general, boron concentration today in our effluent in Israel is not problematical, but all the time we are looking, next please, we're looking and know that without leaching, boron can be accumulated. So this is something you need to be monitor. Next, please. Another problem that we have, it's a boron and sodium chloride at the same time. In that case, you can see the concentration. This is the distribution. I think it was 30 years ago, distribution of chloride or different level of chloride in the effluent. And you can see that we have high level of chloride up to 400 ppm. At the same time, high level of boron. Next, please. And I spent many years in my career trying to solve the question, what happened when you have double trouble, when you have boron and salinity at the same time? The toxicity of these minerals are, the mechanism is different, but still we ask ourselves what happened if you have just 10% depletion of yield because salinity and 10% because of boron. What happened if you combine these together? Are you going to have less than 20? This is what we expect, or higher than 20, 40 or 50%, and your problem is coming to be much bigger. I can say to you today, after many years of research, that when you combine boron and salinity, you get less toxicity than what you expect. Next, please. I would like to finish my talk with water efficiency and salinity. You can see here the range of chloride and sodium in our different water that we are using in Israel. So we have desalinated water. As David mentioned, it's coming to be more popular in the last 10 years. We're using also for irrigation, very low concentration of chloride and sodium. Fresh water, this is a concentration in effluent. Also, we are using saline water, which the concentration of chloride and sodium, it's high. And next, please. One of the questions that we ask ourselves is what to do when you have two different kinds of water, like saline water or brackish water, and you have desalinated water, and you can mix these both together. As I started, the water is water, and the question what happened if you can save water just because you can mix this water together. So we run experiment in the Arava. This is an area that are using saline water, growing vegetables, used to have a lot of pepper. And the curves that you see here, it's the relative yield against relative irrigation comparing to the ET0P, which is the meaning is the water that the environment use. Where one, the meaning of one in this term is the water that the total water that the plants needs in order to grow. And when you are irrigate with saline water, you can see that you get a maximum yield when you irrigate 2 times more water than what the plants need. And the meaning is that you need to leach out all the times a high level of water in the root zone, however, salinity in the root zone, in order to expose the roots to low concentration of salinity. So what happened in the Arava? In order to get this yield, they irrigated twice amount of water. What happened next, please? What happened if you are just supply water with without salts? So you can see here that you need to give the plants only the water that they need. You don't need to wash out the saline from the root zone. You get the maximum yield with a number relative irrigation 1. What happens if you combine 50% of these waters? Next, please. What you get is exactly that you need to supply another 50% water in order to remove the water from the salinity from the root zone. So the meaning is that if you have water with high salinity, you are not going to save water when you are using this water, because anyhow you need to irrigate it more in order to leach out the salinity from the root zone. So you are not saving water. And the last slide, please. And the result, this is the result. This is the concentration of nitrate and the electrical conductivity in the groundwater in exactly the same place, the experiment that I show you. And you can see what happened in the last 30 years, the concentration mostly of nitrate increasing with the time. And it's coming from the agriculture because we are using higher amount of water. Of course, nitrate is moving. This is sand soil. It's easy to leach out the nitrate. And what happened, the concentration of the nitrate increasing with the time. This water cannot be drinking anymore. Actually, sometimes you can irrigate it directly without apply any nutrients, nitrogen, sorry. The last one, please. So I think the take-home message from my perspective is that each type of water has a unique chemical composition, and it should be known by the farmers. And we have the knowledge. When I say we, I It's all the researcher all over the world. Sometimes we still have a question that we're asking, but basically we have the knowledge. You don't need to start everything from the beginning. It's there, it's public. You just need to read and find the solution in your area. The next one is just dealing with the sodium chloride. The next, sorry, can you go back forward? The next take-home message is just talking about decreasing sodium chloride in irrigation water, increase the yield, decrease the leaching factor, increase water and nutrient use efficiency, and decreases environment damage. And I think this is important message. In Israel, we used to use saline water, and today we know that if we have another possibilities, we prefer to take or use water for irrigation, less saline water. Thank you very much.
Thank you very much, Uri. We will have an opportunity later on in the discussion to elaborate. I will again ask the speakers, please be short and to the point because we have a lot of speakers today. So thank you again, Uri. And our next speaker is also from the Volcani Institute, Dr. Shmuel Asulin, who is Associate Director for International Relations and a researcher at the Institute. Dr. Asulin will share research regarding the effects of irrigation with marginal water on soil hydroecological functioning. Dr. Asulin, please. You're muted. Dr. Asulin, you need to unmute.
So shalom. I will share with you some work we have done on long-term effect of irrigation with treated wastewater on clay soils. I am a researcher at the Volcani Center, the Volcani Institute, and many researchers here. We are the most important research institute in agriculture in Israel, and many researchers are involved in different aspects of application or allocation of marginal water sources to irrigation. Please, next. Okay, so we know that the demand for food is increased or will increase, especially for grains. And to meet this demand, we will need to move to much more irrigated agriculture. Irrigated agriculture allows us to increase yields, to increase water use efficiency. And in parallel, the pressure on the freshwater resources is also increasing. You are more population. We need to allocate our freshwater resources to drinking water. So to increase, to expand the irrigated agriculture, we will need to find additional sources of water that are applicable to irrigation. Next. So we identify for the moment 2 additional sources of water for irrigation. One is desalinated water, very low salinity. You heard a little bit about its advantages before, but it needs to be adapted to agriculture because plants need some low amounts of minerals to grow. And the point that I will discuss here today is the use of treated wastewater for irrigation. This water is richer in salts, and it contains also organic matter. Please, next. So the experiment I will show you today is on field plots of avocado trees planted on clay soil in the northern Galilee. We choose orchard because then we don't have tillage, we don't move the irrigation system, and we can look at the long-term effect of application of water quality to the soil and to the plant. We have 2 end members, fresh water of very good quality in a local source, which pH of 7, electrical conductivity, which is close to 1 decisiemens per meter, the sodium adsorption ratio of 0.5, which is very low, and practically no organic content. while the treated wastewater that we have locally has a pH of 8, electrical conductivity of 1.6 decicement per meter. The soil absorption ratio is high, it's or almost 4. And we have also 40 parts per million of dissolved organic content. And we also use these two end members to look at two different mixing qualities between freshwater and wastewater, 1/3 of treated wastewater and 2/3 of treated wastewater. Next. So just looking at the impact of the water quality that is applied by drip irrigation. So we have a very dense soil sampling below the dripper. The dripper is marked in red on the upper picture. And you see how we sample below the dripper, and we compare the soil that receive fresh water and the soil that receive treated wastewater. And in terms of the exchangeable sodium percentage, which is an index for sodicity, you can see that in the freshwater, the values are very low, around 1, and some higher levels close to 2 deep below the dripper, while when you look at the treated wastewater profile, you see that you have very quickly in the root zone already high values of ESP that are close to 6 or up to 7, and that means that soil sodicity will be an element here. Next. When you look at the DOC in the same sampling, you can see that below the dripper that is providing fresh water, there is practically no dissolved oxygen organic content, and the higher levels are close to the soil surface because of litter decomposition, for example, while below the dripper that is providing treated wastewater, you see a very nice plume of very high concentrations of dissolved organic content, and the combination between dissolved organic content and ESP will be the problem of the wastewater application. Next. So looking at a higher scale, you see here the effect of the water quality on the water and salinity regime in the root zone using electromagnetic induction, EMI. This was done with Scott Jones from Utah University. And what you see here is 2 rows of trees, 10 meters from west to east and 35 meters from south to north. So you would see here approximately 10 trees, 5 trees on the western row irrigated with wastewater, and 5 trees in the eastern row irrigated with fresh water. And the level of bulk electrical conductivity that is a response to both water content and salinity show that the trees that are irrigated with treated wastewater are presenting a higher water content and a higher salinity profile. That means that there will be more problematic for water uptake and plant performances. Next. So when we look at the effect of the water quality on the infiltration capacity of the soil, we see that as we increase the component of the treated wastewater, we decrease the capacity of the soil to infiltrate, and this will affect irrigation efficiency. Next. Using this information on the infiltration capacity, we can derive the hydraulic properties of the soil, and we can show that the water quality determine what are the hydraulic properties of the soil, and we will use that in a few slides to derive appropriate irrigation methods. Next. So the combination between higher water content in the soil because of the effect of the hydraulic properties affect also the aeration into the soil. Here we have sensors in the root zone that are measuring oxygen concentration. You see in the plot below all the whole season, all the year, the green bars are the rainfall event, the red and the blue bars are the irrigation period. And you see also the temperature of the soil that increase and peak at DOY of 240, which is August in Israel. So what we can see is that the oxygen concentration in the root zone, in the trees that are irrigated with the wastewater quality, the red line is showing lower concentrations of oxygen in the root zone, and this will impact also on the plant performances. Next. We have here a support to this insight or outcomes from a completely different direction. When we look at the microbiome into the root zone and at the microbial community, we see that the water quality affects the structure of this microbial community in the root zone. And when we look at metagenomic analysis, we find enrichment of stress-associated genes related to high level of salts, high pH, and lower level of oxygen, thus supporting all our physical result and observations. Next. We also have dendrometers measuring the trunk growth of the avocado trees. And for the whole year, we see that the water quality of the irrigation affected the growth of the trees and eventually affected also the yields. Next. So using the observations on the physical properties of the soils, we could try to derive what will be the appropriate irrigation scheme that will mitigate the negative impact of irrigation with the low quality water. So if this experiment, in that experiment, we use what is in the reference box, that means that irrigation was given by 2 drip lines per row tree every 2 days with a discharge dripper of 1.6 liter per hour, moving to 4 drip lines per row and keeping the same frequency and the same drip discharge is the columns on the right. You see that we increase the relative transpiration of the trees. That means we increase the potential for higher yield. And we also reduce the gap between the blue and the red columns, which is the difference between the water qualities. So we can, by observations and monitoring of the physical properties and the chemical properties of the soil, adapt the irrigation scheme that can reduce the negative impact of the water quality. Next. So if we conclude, we saw that irrigation water quality affects soil hydraulic properties, flow processes such as infiltration, evaporation, and plant water uptake. Treated wastewater, when applied to clay soils, because of the impact of the ESP and the interaction between ESP and DOC, decreases the soil hydraulic conductivity, affecting wetting and drying processes, reducing aeration in the root zone, and limiting plant growth. And the complex interactions between the water and the air regimes and the chemical and biological conditions in the root zone affect soil quality and plant productivity. Mixing of water qualities can reduce this negative impact, and Uri talked about that at the level of salinity. But adapting the appropriate irrigation management to the soil and water qualities can mitigate it. That means when we apply a water of a given quality, we in fact create a new soil in the direct environment of the root, and we need to account for the changes in the soil properties so that now we could adapt our irrigation management. So thank you very much, and good luck.
Thank you very much, Shmuel. Thank you very much for being concise. Our next speaker is Dr. Eddie Zitrin, who is also Senior Research Scientist at Volcani Institute at the Institute of Soil, Water, and Environmental Science. Eddie will elaborate on the question of antibiotic resistance along the wastewater effluent soil crop continuum. Dr. Zitrin, please.
Thank you, Adam, and thank you again to the organizers for inviting me. Flip. Next. Flip again. So over the past, go back one, please. The past 40 to 50 years, we've kind of transformed from what was known as an antibiotic era, where we had thought that we won the war against infectious diseases, Flip, to the current state, which has been defined as a post-antibiotic era, which in effect is a pandemic-like situation where we cannot treat infectious diseases because bacteria are not only resistant to antibiotics, they can be multi-drug resistant to a wide array of antibiotics and thus can't be treated. And this phenomena stems from both selection due to exposure of bacteria to antibiotics, and also to a process known as horizontal gene transfer, where bacteria can actually transfer genes through various mechanisms from one to the other. And in the end, we end up with a situation, as I mentioned, of bad bugs and no drugs. Next. Now, the phenomena of clinical antimicrobial resistance is quite well known in both the scientific community and the general public, but there's a rising understanding that antibiotic resistance is a more holistic problem, which is associated with many different types of environments. And here we'll focus on wastewater treatment plants because they actually receive all of the fecal communities and residual concentrations of antibiotics that come from the sewage. Now, one thing that's important when we look at this environmental dimension, as I mentioned, genes can be transferred between bacteria, and therefore we not only have to look at the bacteria and specifically bacterial pathogens as a problem, but also as the genes. And these genes have been characterized as emerging contaminants or contaminants of emerging concern. And therefore, often, in addition to looking at the bacteria themselves, we can use PCR-based approaches to monitor and quantify the genes and as a mean of source tracking. Flip. I think I don't have to elaborate on the issues of treated wastewater irrigation in Israel. Flip. But it's important to know that this isn't just an Israeli problem, it's a problem that's going to increase due to climate change and increasing population in parts of the world that currently don't face water shortages. Flip. And David's done a good job of introducing the standards that are used in Israel and other parts of the world. And when me as a microbiologist look at these standards, we obviously see fecal coliforms or sometimes E. coli, but there's a growing understanding, and there's been some discussion within European regulation boards that we not only have to look at fecal coliforms or E. coli themselves, we also have to look at antibiotic resistance. Flip. And I'm going to give a very, very short taste of a number of studies done in our lab and try to integrate them in the end. And this is a study done specifically looking at multi-drug resistant E. coli in secondary effluents from wastewater treatment plants, done by Tamar, who is a master's student. And the take-home message of this is, when we look at a large array of multi-drug resistant bacteria, we can see a trend, and you can kind of look at the upper part of the figure, where the earlier the different types of antibiotics had been introduced into the clinic, the more resistance we see. So basically, the later the antibiotics, the newer the antibiotics are, the less resistant strains we see. And this is kind of an indication that as time goes on, if we continue to use these antibiotics, we're going to be seeing more and more traces of resistant bacteria that are going to be spread into the environment. And of course, within this One Health perspective, this can be a very big problem. Flip. This is emphasized also when we look at the genes of these bacteria, and specifically plasmids that are these mobilizable units that I talked about before. And when we look at these plasmids, not only do we see that they can contain resistance to a wide array of genes, they also are sort of like telephone apps where they can acquire and transfer genes. And therefore, a plasmid that currently doesn't have a resistant gene for a specifically a problematic antibiotic can acquire these genes in the future. Flip. I mentioned up to now, I was talking about culture-based methods, so isolating E. coli or coliforms. Now I'm going to a study done by Roberto Morano, who was a PhD student in the lab, who did a very, very large survey of both wastewater treatment plants, effluents, soils, and crops irrigated with these wastewaters, treated wastewaters, looking at a wide array of antibiotic resistance genes. I won't go into all of them and why we selected these specific genes, but what we can see is that there are much higher levels in in treated effluent samples than in freshwater samples. And if we try to extrapolate how much of or how many of these genes in copy numbers are being transferred per square meter to irrigated fields per day, we're talking about huge amounts of these genes, which can be problematic. Flip. But then, as shown in this publication in Water Research, when we look at the soil and the crops, we actually see that most of the genes were below detection levels or detectable but not quantifiable. And what was very interesting was that we didn't see strong correlations between the abundance of these genes in the water that was being used for irrigation and the abundance of genes in the corresponding soils and crops. So it appears, Flip, that soil and crops are somehow ecological barriers to transfer of these bacteria and genes, and we have a number of other studies that support this. However, and this is a publication that was recently published in Environmental Science and Technology, one of the things that we can see is when we irrigate in experimental systems with multi-drug resistant E. coli strains, and you can see in the top graph, it might be a little complicated, we do see that these strains dissipate over time. So over time, they don't survive in soil, or at least they go below detection levels. But if we enrich for these samples using specific media, we find that often, even 2 weeks after going below detection levels, we can actually detect them. So they're sort of below the radar, and these are phenomena that we weren't aware of before. And I won't go into all of the details. We targeted a number of different study sites and found that this phenomena was specifically evident in soils that were irrigated with secondary, so low quality treated wastewater, but it wasn't as evident in soils that were irrigated with high quality tertiary treated effluent. Flip. I want to bring your attention to two very interesting studies that try to combine not just looking at coliforms and fecal coliforms, but also the aspects of antimicrobial resistance. This was done within the framework of a European cost action. And what we did in the left slide was to not only look at fecal coliforms, but also look at fecal coliforms that are resistant to a third-generation cephalosporin, so a relatively new type of antibiotic. And what was interesting was that we saw that the level of fecal coliforms coming into wastewater treatment plants, and this is a study that was done by 34 different groups in 23 different countries around the world, the variation in total fecal coliforms was relatively low, but the variation in cefotaxime-resistant coliforms was very, very high. And this is something that we tried to kind of take home in this publication, is that in addition to these standard methods that are used for looking at fecal coliforms, we should also measure antibiotic resistance, and specifically resistance against these emerging antibiotic compounds that provide a lot more insight. The second study on the right was looking at antibiotic resistance genes, and we basically tried to say that not all antibiotic resistance genes are created equal. We have some that are very prevalent in the environment, some that can be persistent, but some that have been characterized as hazardous, and these are the genes that can transfer between bacteria and that are found in clinical pathogens. And if we want to use these PCR-based approaches to look for antibiotic resistance genes, we have to really be able to make standards of which genes to look at and what the thresholds are to consider if they're dangerous or not dangerous when we irrigate with the treated wastewater. Flip. So to summarize and try to present some recommendations from this, we can say that wastewater effluents, and especially secondary effluents, contain relatively high levels of antibiotic-resistant bacteria and antibiotic-resistant genes. Nonetheless, we found that soil is a strong barrier to antimicrobial resistance dissemination, and resistance levels in soil and crops are generally below limits of quantification. Saying that, though, we find potentially pathogenic resistant bacteria can persist in soil under the radar when low-quality effluents are used. And as recommendations, we can say that current wastewater use criteria appear to be suitable in regard to antimicrobial resistance, but efforts should be made to upgrade secondary to tertiary treatment where possible. I'm not sure that we need values that are higher than the standards that we're using today. Another thing that's important is routine surveillance of fecal coliforms or E. coli and effluents, which should be supported by periodical assessment of antibiotic-resistant fecal coliforms and E. coli, and also assessment of antibiotic-resistant genes. And with that, I'll finish. I just like next flip to thank, of course, the students and postdocs who collaborated or who worked on the research, our collaborators from Hebrew University and the Ministry of Health, and of course, European partners and the funding agencies. And thank you again for listening.
Thank you, Eddie, very much. And now we turn to something slightly different. The last speaker of this session is Professor Yoram Kapulnik, who is the Executive Director of BARD, which is the US-Israel Binational Agricultural Research and Development Fund. Professor Kapulnik, you have 5 minutes, please.
Okay, thank you very much. Can you hear me there? Can you hear me?
We hear you, yes.
Okay, thank you very much for inviting me here to this fantastic session. And I would like to thank the scientists for the enlightening talks, really interesting data. It's really very, very important. And my name is Yoram Kapulnik, and I'm the director of BARD. And as was mentioned before, BARD is owned by US and Israel jointly. This is a funding mechanism, and I'm going to devote the next 5 minutes or 4 minutes to explain a little bit what we are all about and where the opportunities are. So next, please. In the next slide, I'd like to highlight that BARD is a competitive funding program that supports collaborative agriculture R&D in areas of mutual interest to the US and Israel. This is a kind of a sharing effort of the two nations to science, innovation, technology, and development. And BARD is the stockholders of BARD is the Israeli Minister of Agriculture, of course, and the Israeli Minister of Finance here in Israel, and in the US, US Department of Agriculture, and of course our natural partners there are ARS, which are part of our activity. Next, please. Now, BARD is funding research in a very wide area. A lot of topics are there. Actually, we covered a lot of the important topics in agriculture. We have 8 panels, starting with animal production, animal health, crop production, crop health, food products, quality, safety, and security, environment, renewable resource and renewable resources, agricultural engineering, and agricultural economics. And that actually BARD was funded, if you notice, in 43 years ago. And we actually very proud to present some of the numbers which are associated with our activity. Next, please. In the next slide, you'll be able to see that over the years, more than 900 American principal investigators enjoy the activity or the funding from BARD. And in Israel is more than 600 principal investigators. And most of the activity you can see here on the map, the number represent the number of awards which came from each state. So we actually are relevant, or a lot of agriculture research was supported by BARD in very different, actually I know exactly 47 different states. And we are very proud that we are actually spread around the US. Over the years, we were able we received more than 5,000 proposals, out of which we supported 1,300 grants. All in our program, we actually invested more than $300 million. Next slide, please. Now I'm very happy to share with you some of the information which we collected in our assessment. It was an impact assessment which was evaluated by international committee. So the invested amount was $315 million. But the most important parameter is that our ROI, if you measure what is the economic contribution of each dollar, you'll be able to see that each dollar actually contributes $16. It's a very enormous number, and it's actually being directly related to the fact that this is a very competitive grant, and if you choose the best of the best, you will be able later on to receive some impact in the market. Another parameter which is very important, most of the scientists that collaborate, 82% of them continue to work together beyond the duration of the project. It's a very dramatic kind of matchmaking, and these people are working together, different scientific funding frames that they are succeed to receive. A lot of publication, more than 5,000 publication, commercial engagement, new jobs, patents, breeding rights, and regulations. And this is really a lot of information. I really want to encourage you to take a look in this assessment, which is on our site, please. Next slide, please. Over the years, I'm talking about 40 years, and here I'm coming closer to the topic of the session. We were able to support 130 research projects with water and 4 workshops. As a matter of fact, one of them will be this year, which was funded already. And so the total investment of BARD already, again, we are a funding mechanism, is more than $40 million, which was invested in topics which are relevant to water. Part of it, and here it's the dissection, it was plant-water relation, irrigation, soil water quality and quantity, water purification and wastewater management, economic evaluation of policies. And this is a very wide array of specific topics that were funded by BARD already. I would like, next slide, please. So
we really need to finish.
To convince you, in a minute, convince you that BARD is a worthwhile mechanism. It belongs, both nations are invested in that. I just want to let you know that recently we developed another mechanism. We call it BLIVER, or BARD leverage. And this is a bilateral collaborative models which take further the research to the market, to commercialization. Again, I don't have much time to expand on this program, this new program. I invite you to take a look in our website. Next slide, please. You can see here our address, and we will be very happy to assist you in any request to go further in any objective of your science and technology. Thank you very much for your attention.
Thank you very much, Professor Kapolnik, and I urge the audience to really make use of this mechanism and contact the BARD if there is any, if you see any potential And now before we turn to the second session of the day, I want to remind you all that you have a Q&A button on beneath the presentation where you're invited to pose questions to our panelists who will do their best to answer throughout the webinar. There will be a discussion at the end, but given that we won't have too much time, you will also have this possibility to post questions in the Q&A box. And now we turn to the second session of the day, which will introduce you to innovative technologies and solutions applied in agricultural irrigation in Israel, developed in Israel. Now, reusing water is great, but doing it efficiently is even better. So I would like to invite two representatives of Netafim to talk about innovations in drip irrigation. The first one is John Farner, who is the new Chief Sustainability Officer, and Ido Ra'anan, who is Circularity and Sustainability Global Leader. Gentlemen, the floor is yours, please.
Ido, go ahead.
Yeah, thank you, Adam. Please go to the first slide. Yeah, so I don't need to introduce myself, but John, do you want to say a few words?
No, our time is short. I just want to thank the folks from Israel as well as the United States for having us on today. The presentation has been fantastic, and I'm excited for Netafim to be a part of this and sharing our solutions. So, Ido, floor is yours.
Yeah, okay, thank you. Next, please. So Netafim was formed and founded some 56 years ago in the Israeli Negev desert in the kibbutz called Hatzerim, which we see the aerial photo of the kibbutz, and we can see how we can make the desert bloom, putting the right amount of water for every plant. And we can see the bottom picture how you can use efficiently the water and put it exactly in the right spot, enable the agriculture where it wasn't possible before. And it is not only in the Negev desert, it is around the world. Next, please. So in the last 2 days and in the last few lectures, you already heard about the Israeli infrastructure of wastewater treatments, and the numbers are quite the same. I did want to emphasize about the national infrastructure that allows the most distant farmers to have a high quality of treated wastewater, and that will become handy in the next slides. Next, please. So here we can see some kind of schematic illustration of a field. I do not have the cursor here, but you can locate in the center of the picture the system head. The water, the treated water, is coming to the system head, maybe pressurized with a pump and going through a filtration system, whether it's going to be disk filtering or media sand filters. We need volume of filtering, not only surface. We've got fertilizer tanks, mixing system and controller. And then from there, it's going to the sub-main. This is the white line in the center of the picture that distribute the water to the drip lines, which can be either subsurface drip irrigation on the left or surface irrigation on the center. Usually with the treated wastewater, we do not use the sprinkler system that we can see on the right. Next, please. So this is a sketch of the system, and I wanted to highlight here just a few important elements when we are talking about the wastewater. You can see the direction of the flow of the water from the pond or from the water source on the left, going through the pump. And the first station that I will stop is the filter. With the years of experience dealing with wastewater or water for irrigation in general, we understand that we don't need more than 120 mesh filtering system. Sometimes we can use even less. It depends on the irrigation system, which I'll speak about in a few seconds. Next, we can see in station number 13. No, not next. Go back one slide. Thanks. So we can see in the upper part of the slide, number 13, this is the controller and the mixing system. This is a crucial part for the next slides. So that's just bear that in mind. And when we are going to the actual drip lines, where we can see number 18, it depends whether it's going to be a seasonal drip irrigation, which in the end of the season will be collected and sent for recycling, or being collected and reused in the next season. So the system that is being reused for many years need to maintain in a strict regime of flushing to prevent building up of material that was not filtrated but built up inside of the drip lines. We can see the flushing valve at number 21 on the bottom left. Next, please. So here we can see, don't play it just yet. We can see here a nice video that exemplify the work of a dripper. The dripper, we do not see it right here, but it's got an integral filter on the back that prevent the biggest or the bigger particles to enter. But each particle that will enter the emitter, the dripper, will eventually go out due to the special mechanism of the labyrinth. And now we can play the video. So we can see the water entering the labyrinth, filling it up. And you can see the turbulence inside of the dripper. This is a very sophisticated, it looks simple, but it's very sophisticated system that allows the particles, as you can see now, going through it and not building up, not stacking inside, and eventually being emitted out of the dripper and out of the drip line. Next, please. Next slide, please. Now, all of that experience brought us to the point that we are seeing an opportunity to use the effluent of dairy farm, using the controlling, the sensing, measuring the electric conductivity of the water, and understanding the amount of nutrients inside it. Next, please. So in one of the slides of the former speaker, we saw that we use it, the manure in order to closing the loop and fertilizing the field. But as we can understand, this is a messy business, and we are looking for something more hygiene and elegant. So next, please. We came to a circular solution to put the material, the effluent from the dairy farm directly into the drip lines, treating it, monitoring it, diluting it and mixing it with fresh water. And the results were saving both nutrients, energy, water, greenhouse emission with, like in every circular economy solution. And in the next slide, we can see a nice video, 1-minute video that shows this solution. So next, please, and please.
To be able to grow a forage crop, you need water. If we don't have water, we don't have a crop. So when we talk about sustainability, this system really closes the loop. So first, the cow will eat its feed, process the feed, and then waste is produced. That waste gets flushed out through a wet dairy system, which means they actually wash the pens down several times a day to keep it clean for the cows. That water and nutrients is brought into holding ponds, where it's passed over an angle screen or into an area where solids are separated out. From that solid separation, it goes into either your secondary or tertiary pond. It's more of like a tea. From that tea, it passes through some pre-filtration into the system. At that point is where we blend the effluent water and the fresh water to make the desired blend that we need to go out to the field to be utilized by the plants. From there, it goes out in the field on a fully automated drip system that's put out exactly when the plant needs it and correctly across the field. From there, you grow the crop, you then harvest it and take it back into the cow. So it's a complete circle of everything being utilized on the dairy.
It's a happy plant. Happy cows, happy plants.
Yeah, so nice one. And with that, we'll finish our presentation. We will, of course, be happy to
be able to grow a forage crop.
We will be happy to answer any question that may arise. And we've got 1 or 2 minutes more. So John, if you have any closer.
Yeah, just real quick. I think it's important to note that that example that we just saw in the video is in California. So it's a great example of this rich history of Israeli technology that Netafim has being implemented around the world, specifically in this case in California, which is where the first time that this specific process took place. So it's a great example of the coordination and connection of public-private partnerships and this Israeli technology being utilized in the United States.
Okay, so thank you very much, Ido and John, for this illuminating presentation. And now I would like to give the floor to Dr. Oshri Rinot of Suplant, who will present other smart irrigation solutions. Oshri, please.
Okay, hello everyone, and thanks for the organizers for inviting me for introducing Suplant technology in this fascinating event. Next, please. Next. Okay, so fewer than 2% of farmers use computer models to inform their irrigation decisions. And my main goal in this presentation will be to convince you that the rest 98% are actually wrong, and under conditions of climate change and water scarcity, these models are really essential. So our platform is first based on sensors. The sensors are located on the fruit to measure fruit growth and development over time. On the trunk, I will see some example later. Leaf temperature sensors that are measuring the soil moisture to estimate the water content in the soil, and meteorological stations that measuring all the weather data. In addition, we are connected to a weather forecast system that is also providing major added value. So our data is collected every 10 minutes, so it provides high resolution data in real time, providing powerful platform for smart irrigation insights and actions. Next, please. So this is a fresh from the oven example from the last week of an olive orchard here in Israel that is demonstrating the ability of the plant and soil sensors to indicate the water status of a specific plot and to provide accordingly some relevant irrigation recommendations. So this figure is illustrating the dynamics of the soil moisture in the soil, and the soil moisture is measured at 2 different depths across the soil profile, and the distribution of the soil moisture across the soil profile may help us detect some water infiltration problems that may be associated with treated wastewater irrigation, as explained before by Dr. Asuli. So we can see a trend of a sharp increase in the soil moisture, and then the soil moisture is gradually decreasing over time. The sharp increase is due to irrigation, and the gradual decrease is when the soil is drying between the irrigation until the next irrigation is conducted. And you can see that the increment between irrigation and irrigation is about 2 or 3 days. And we may ask ourselves if it's good or not good for the plant, if the plant is okay with this or not. And we cannot conclude any answer from just looking on this figure. In addition, when speaking about treated wastewater irrigation, when the salinity is a major concern, as time is passing since the last irrigation, so the water are evaporating and the concentration of the salts in the soil is increasing, and the overall soil salinity is increasing, and we are actually maybe under salinity stress. So, so as a result, we just need to ask the plant what he thinks about this irrigation, if it's good, if it's not good, and we do not want to wait until the end of the growing season to see that our obtained yields are lower than expected. Next, please. So here we can see the plant response to the irrigation. The brown lines here are indicating the trunk radius. The trunk is shrinking at days and swelling at nights due to physiological mechanism associated with the water uptake and water loss during a 24 hours period. And we have here two main parameters. The first parameter is the trunk growth rate, marked by the white arrows. We can see that just after the irrigation, the trunk growth rate is positive. And then as time is passing since the irrigation, the growth rate is decreasing and becoming even negative until the next irrigation is conducted, and then it becomes positive again. In addition, the second parameter is called MDS, maximum daily shrinkage. It's the difference between the maximum and the minimum trunk radius on a daily basis. And again, we can see that the MDS values just after the irrigation is low, and as time is passing since the irrigation, the MDS values become high. So high MDS value with negative trunk growth rate are actually telling us that the plant is under stress. And the conclusion is just that the farmer needs to increase the frequency of the irrigation. So next, please. In order to well translate this signal to precise irrigation recommendations, we must also take into account the climatic conditions. And when I say climatic conditions, I mean to the detected data in the meteorological station and the forecasted data. So the most relevant climatic conditions for irrigation are the VPD, vapor pressure deficit. It's actually the driving force of the water from the plant to the atmosphere. And of course, the evapotranspiration, which represents the potential water loss from the plant, from the soil. If the VPD is too high, so the plant may be under stress. And if the stress is occurring at a critical stages during the growing season, so it may have a very high and negative impact on the obtained yields. And this is very, very important under the global warming responsible for higher frequency of such heat wave events. But there's something we can do. We can actually, as a plant, we can actually be prepared for such events and irrigate the plant before such event is expected. And our system is providing precise irrigation recommendations regarding the amount needed for irrigation and also the timing. The timing is very, very important. Next, please. So in addition to the plant and soil sensors, the measured and forecasted weather data, our irrigation recommendations are based on broad database and a vast experience. And consequently, the recommendations can be adapted to specific irrigation requirements of different crops at different seasons in different parts of the world and at different phenological stages along the growing season. Next.
Oshri, we need to finish in 2-3 minutes. So
Okay,
please.
no problem. So
thank you.
As written here, we trained AI models on this data to find a path to maximize every plant's daily and seasonal potential by optimizing irrigation amounts and timing. Next, please. And this is actually the final result of the algorithm. It's a well-specified irrigation recommendations and alerts. You can see in the figures that the irrigation amount are not constant and they are dynamic. They're dynamically changing during a week. And this is illustrating the flexibility of the algorithm and its ability to be adjusted to the changing growth conditions. In addition, the system is continuously monitoring the data obtained from all the sensors and alert the user for any detected or forecasted change in the plant, soil, and weather data. Next, please. So just to summarize, this is the process of the technology. First, the sensor measure plant data every 10 minutes. Then the data collected from the plant sensors is uploaded to the cloud. In the third stage, the plant unique algorithm analyzes data and translates it into actionable agronomic recommendations and insights. And in the last stage, farmers are provided with precise, real-time and forecasted agronomic insights and irrigation recommendations via the plant mobile app and desktop platform. Next, please. Just to example how actually the supplant technology is increasing the obtained yields with lower amounts of water. This is an example of apples from Australia. Next, please. And this is an example of oranges from South Africa. And again, the same pattern, more yields with less water. Thank you. Next, please. Thank you very much for listening, and I'll be glad for any questions. Thank you.
Well, we'll have a discussion at the end, or the audience can post questions at the Q&A. So thank you very much, Oshri. This was very, very interesting. Now we have the last speaker of today, Dr. Victor Al-Khanati, who is Senior Research Scientist at the Volcani Institute. Victor will show us how to implement precision irrigation using drone tech. Dr. Al-Khanati, you have the floor, please.
Thank you very much. I have the honor and the opportunity to talk with you on 3 different subjects in the next 10 minutes. Precision, irrigation, and drones. 3 things that they come together in what we call FLIP. Next, what we call together as precision agriculture, or digital farming, or smart agriculture, which is actually the new thing of the new thinking about agriculture, of sites specifically managing the resources. Next. When we're talking about precision in irrigation, I want to convince you, or to let you know, that there are systems that they can convey water in a spatially variable way, where we built maps, like the ones that you see on the right bottom of the image, and we can apply different amount of water in each part of the field according to the need. Next. Even with systems that they are not center pivots, Netafim has developed a system that uses a controller or a number of controllers with a number of valves that can apply water individually in different small areas. So again, we have site-specific irrigation or precision irrigation. Next, please. In order to do that, we have to acquire data. There are a lot of platforms for acquiring data. Can you please start the videos? Thank you, all of them. We have satellite platforms that you see on the left, or ground platforms like the ones you can see on the bottom right, or we have drones or UAVs that together today have the opportunity to give us data that they are in extremely high resolution. Next, please. The most maybe important or a new thing that has been done the last years are the nano satellites in the space area, where the nano satellites that you see on the bottom right part, that they weigh about 4 kilograms, they are trying, and actually they are replacing in some cases the big satellites that you can see on the left part of the image. They can provide us information through constellations of satellites. In this case, for example, there are more than 200 satellites, nano satellites, that they provide a map of every centimeter of Earth, of the surface, every day. Next. Probably
Dr. Khanati, can you try to improve your audio? We have some trouble hearing you. There's a background noise.
Is it better now?
No, it's worse.
I'll come closer together. So what we can see here is maybe the most flexible platform of acquiring data, which are drones or unmanned aerial vehicles. We have a number of them. Some of them are winged aerial vehicles, like the ones that you can see on the bottom left or top right. We have helicopters like the one in the center and the top, or drones. Next. Those become platforms more and more popular. You can see here the number of articles and papers on UAVs. The last years, they have an exponential growth. And actually, there are some people who predict that by 2030, there will be 1 billion drones flying around us. Thanks. Now, what do we do with these platforms? In order to manage irrigation, the conventional thing was to use the environment as the input of how much to irrigate, like pan evaporation models like Penman and Pink, or even by measuring the soil potential, water potential. Next. Then this management got more sophisticated by asking the plant how it feels, like the last lecture that we heard. We have sensors that are placed on the plants, and the plants can say how they feel in terms of water status. But all this is point measurements. And what we want really to do with the drones is to make maps and measure the spatial variability of this water stress. Next. In order to do that, we exploit the fact that water is evaporated by the leaves. Next. And by having the water evaporated by the leaves, then the energy balance between the leaves and the surroundings changes. Next. We can measure this energy balance, next, by using the electromagnetic range spectrum, by looking at the visible spectrum, which does not really give much information, next, but also by looking at the near-infrared and thermal range, where there is a lot of information about the energy balance between the leaves and the environment. Next. Looking at the near-infrared region of the spectrum, we can see features of the plants that we cannot see with their eye, usually by stresses that they are not specific. So we are not really certain if it's a stress which is caused by water stress or a stress which is caused by another cause. Next. When we're looking at the thermal range, we can see the temperature of the leaves, and the temperature of the leaves is a direct thing that is caused by the evaporation through the stomata. During water stress, which can be due to salinity or another cause of water unavailability, the temperature of the leaves rises. If there is enough water available and enough evapotranspiration, then the temperature of the leaf drops, is lower. Next. So if we take this as an example, here we see a field where half of it is well irrigated in the bottom part of the image, which is dark, and the higher part of the image is light, which is a higher temperature. So we can very easily distinguish between different temperatures in the field, although we cannot see it with bare eyes. Next. By doing that, we can also see individual drippers that they emit. Like in this example, where in the bottom part of the thermal images, you can see the dark plants which evapotranspirate much more than the surroundings. Next. You can also see clogged. Next. You can also see clogged laterals, like in this cotton field, where in bare eyes you cannot really see anything. Next. But in the thermal image, you can see the high temperatures, the light colors here, which are all laterals that they are under irrigation. Next. We can use this technology in order to detect leaks of irrigation systems. This is just an example of the result of research where we found that in vineyards, in olives, in different areas, in different times, we can detect automatically more than 80% of the visible leaks. Next. But the most difficult part is to evaluate the water stress. Next. In order to manage irrigation. Next. In order to do that, we have to use the canopy temperature next and normalize it to a value where the farmers can use it, stomatal conductance next, or relief water potential. This is a parameter that the farmer can use to manage the irrigation according to existing agro irrigation models. Next. In order to do that, we have to measure the environmental parameters because the canopy temperature is not directly translated to water stress. Next. But if we measure leaf temperature, air temperature, net radiation, and all the parameters of the environment, we can calibrate the temperature and translate it to water stress. Next. If we do that, then we can map the leaf temperature that is measured by the drones or other high-resolution imaging techniques and make maps of the water stress or water status variability, as you can see here in a single cotton field during 3 different periods of time in 2011. Next. Now, if we know also the agrotechnical model of how to irrigate according to the parameter that is yielded by the thermal imaging, which is the central part of the graph that you see here, then we can translate it to irrigation management. And we have shown that using this technique, we can increase significantly the water use efficiency in cotton fields. Next. We've shown the same thing in potato fields, like the one that you can see here. Here we use the drone with the thermal camera, and you can see in the right part of the slide the different colors which indicate different temperatures, which are because of different irrigation regimes. Next. So I think that in those few minutes I have showed you that next, drones can map plant water status and spatial variability. Next. But thermal imaging provides a direct plant feedback on water stress. Next. These water stress may be caused from water deficit or salinity or diseases or any other causes, but they are expressed through the leaf temperature. Thank you.
Thank you, Dr. Al-Khanati. I mean, that is amazing. I mean, all the presentations here, it's just a combination of modern and high tech. It's always fascinating how you can apply that. So I want to thank really all the speakers of today for the enlightening presentations. And now, without further delay, I'm very pleased to hand over the moderation to Dr. Clinton Williams, who is a lead research soil scientist, irrigation and water quality research at the Arid Lands Agricultural Research Center of the U.S. Department of Agriculture. Dr. Williams will lead a moderated Q&A discussion with our speakers. Clinton, please.
Thank you. We're a little bit over time, so at this point, I think we will plan on the Q&A session going until about 1:15. And with that, I'd like to say that this has been a great session. We've been able to learn a lot. I think more than anything, this has answered quite a few questions that growers here in the United States have with regards to accepting wastewater reuse for agriculture. For the most part, you've touched on the main topics of salinity and potential contaminants and how to deal with the issues that surround using wastewater for irrigation. We have a few questions that have come in. And the first one is, when Israel first proposed the reuse of wastewater for irrigation, did Israel anticipate concerns about salinity and nitrates? And if you did, were you able to come up with management practices and strategies that were able to address the issues, or have you had to change on the fly as you've been using this wastewater for irrigation? So I'll open that up to anyone.
Clinton, I think I'll take it. It's Danny Greenwolton, Israeli Water Authority. I'll start with chlorides. Definitely, we expected there to be a problem. The work on salinity from Riverside in the 1950s was very well known. And I think there were three different approaches taken during the time. The first one was adapting the crops and picking the area. In the first years where treated wastewater was used, there was a high salinity, there was no regulation. So you would pick a crop. So you wouldn't pick avocado like what Dr. Shmuel Sulin showed us earlier on, because we know it is sensitive. But you would pick cotton, and you wouldn't pick it on an area where you would have an aquifer, but you would find a area where you don't have an aquifer and you have good drainage. The second stage was source prevention. Let's find where the salinity is coming from, and let's make sure it doesn't get to the wastewater and doesn't get to the treated wastewater. So Professor Uri Yirmiyahu talked beforehand about special detergents in Israel, which aren't based on boron and not based on, less based on nitrate, on sodium, sorry. We have special industrial regulations, so you don't pour chloride and sodium into the system. And we managed to reduce the levels. And the third stage is what we talked on the first day on the integrated water management of Israel. Since 2005, we are using large-scale use of desalinated water, seawater. And the desalination is very, very low. We come into the municipalities with a chloride level of 20 ppm. And then the town would add 70, 80, 100 ppm of chloride. But I'm coming out with treated wastewater, which has lower salinity levels than the natural water in Israel, surely of the Sea of Galilee. So we've evolved during the years with our solutions, with better regulation, with better options how to deal with it. And crops that at the beginning we couldn't dream of growing, again, avocado. No one thought about having avocado in treated wastewater. Today it's a practice because the qualities are quite better. Nitrogen, I'm less aware of the history. Maybe one of the other panelists could join in and share his experience.
Okay, if I can add a point, yes, of course, it was expected that salinity will be a problem. But what happens with the years and with the research was the surprising impact of the interaction between the organic content and the sodium, and this was not expected and was discovered during the years of observation and monitoring and all the work that has been invested in the adaptation of this water quality to agriculture. So there is things that you can expect, but there is things that you learn also in the journey.
Okay, maybe I can just add that as for nitrates, the farmers have to take into account that nitrates are in the effluent and have to use less of adding of nitrates.
So I have one other question came in with regards to salinity, and that is, if you have saline problems, what methods do you use to give yourself a leaching fraction or to move the salinity out of the root zone?
I know that farmers in some regions have to flush once in a while, make flushing of the earth to get rid of salinity. It's a practice used.
Yeah, you increase the amount of irrigation, as was mentioned previously today, and you flush it out. It means you have to work with the soils, which you can have good drainage, And we talked earlier on about the influence of organic matter in the treated wastewater, which could cause drainage problems, which is one of the reasons why we're pushing for higher quality treated wastewater, not secondary, but have more advanced with lower contents of organic matter.
I just want to add the problem of sodicity, and I think Shmuel talked about it. So salinity is not just to remove the salts. Sometimes in heavy soil, it's coming to be sodicity soil, which is big trouble. So it should be avoided to have a sodicity soil.
You need to take into consideration also, on top of what Danny just mentioned at the beginning of his first answer, that on top of the regulation on 2011, actually, we kind of changed and put some different tariffs on industrial waste that is going into the wastewater treatment plant. And that for is not just in terms of stuff like criminal law and other regulation, but the financial issue and the higher tariff for industrial waste that is going into the wastewater treatment plant actually decrease the amount of those substances in the wastewater treatment plants, and of course afterwards in the effluents that are going to the farmers.
Thank you. So shifting gears a little bit away from salinity into other water quality issues, can you share the approach that Israel has for thinking about water quality standards for pharmaceutical benchmarks for agricultural reuse? And if they're proposing any limits or if there are any regulations regarding pharmaceuticals in wastewater from reuse?
Yes, well, a few things have to be taken in account. First of all, people know how to, humans know how to make 130,000 chemicals. And the CECs we find in many places. Actually, if you look at the quantity of irrigation in California itself, it's much greater than in Israel. Just our percentage is bigger. But all the research down till now showed no risk of the CECs and the concentrations we find via irrigation to the public health. So I think there's, as there's no regulations around the world, neither in United States and neither in Europe, more research is needed to be done to find out if there's any risk. I can say that we made a health risk assessment to one chemical named carbamazepine, and we showed that the concentrations that are in the produce, and even if you eat all your life rich vegetable diet, there's no harm expected. So more research needs to be done, and for now we don't know about any hazard to public health.
Maybe can I make just one short point on that? They talked about boron and reducing or preventing the use of boron because of its long-term effects. And I think that this isn't necessarily connected to treated wastewater irrigation, because they find carbamazepine in groundwater, it's non-degradable basically. And therefore, I think from a holistic perspective, if there are alternatives, for example, to pharmaceuticals that don't degrade in the environment, then they should be used. So just thinking long-term, I think that might be a way of looking at things, and instead of trying to solve a problem, to try to prevent the problem from the start.
So another question with regards to remote sensing and trying to figure out how to do irrigation based on, say, drones or remote sensing. At this point in the United States, there's a lot of effort being put towards that. And the efforts have moved from, they've gone everywhere from using pin and Monteith and weather stations to trying to use satellite data, but you have to have much larger like field sizes to any sort of like sensors going through the field. Do you see that the use of sensors for irrigation scheduling has a real potential on like an ongoing basis, or is it something that we still have like a lot of technical difficulties to overcome at this time?
I can take this. The use of sensors and remote sensing is not going to change the way that irrigation is managed today as a whole, because what we get from remote sensing and sensors is the feedback from the plant, and actually the amount of water that we have to apply is also a function of the climate. Human motif will be there always. KC, the cost coefficient will always be there and will have to be evaluated properly. But remote sensing will give the additional feedback in order to fine-tune the irrigation according to how the plant has reacted to the previous application of irrigation that has been given.
I would add to that I think that drones, for example, will be really useful in the next few years in terms of specify and do some more accurate herbicide and pesticide use. So with that and with the remote sensing and the sensors, you'll have more specific and more accurate data regarding what actually the plant need. And then you'll use less pesticides and herbicide from one side, and the effect on the soil and on the ground and the whole environment will be much less. Although nowadays, at least in Israel, we don't have any regulation regarding this. And this is something that we need to bear in mind and put some of our efforts on. Thank you.
Maybe I can, excuse me, maybe I can add to Dr. Fanati's words that after having the sensing and the map, you need to close the loop in order to react to that. And we are definitely going in that direction, enabling the farmers to react in more accurate resolution in the field to the different need and uniformity and the opposite of uniformity.
Yeah, just to strengthen this point, you know, drones and sensors and all these things are technologies, and technologies are developing much more faster than understanding processes and understanding what needs to be done after you have this data. So the key point is not gathering the data. The key point is understanding the processes so that the algorithms that are using this data are guiding us toward the good decisions. And for example, if you have dendrometers that are measuring trunk growth and you have values like MDS, and you don't know exactly how this is correlated to the process that is into the tree, you can reach wrong decisions just by using the same type of data that you are gathering with the technology that is more and more available now these days. Thank you.
So we've also had some questions come in that are kind of related, but not, there's like two different prongs to the question. That is, the first one is, because of the water reuse and the way that of the treatment, you provide the water to the growers, the reclaimed water. And the question is, how much does that cost? And with that cost, can the farmer still remain competitive with regards to international markets for agricultural products? And the second part of that is as far as the treatment is concerned, there was someone who was asking a question about treating for boron and how exactly you treat for that, or do you just do prevention of boron?
I'll take the first part of the prices. Prices of treated wastewater in Israel for the farmers run about between 23 to 27 cents, American cents per cubic meter. That's about half of what potable water would cost. The way we get to these lower prices is because the infrastructure is subsidized by the state based on external benefits, which have nothing to do with the agriculture, have to do with the farmers, have to do with the ecology and with public health. And so we managed to have the treated wastewater at an attractive price for the farmers. We do have a question in Israel, and this is something we're talking about a lot in the last few years, of what is the ability of the agriculture to pay for water prices? How can it compete in the Israeli market? How can it compete in the international market? How can it compete in importing, subsidizing for agriculture? But I think this price range encourages the farmers to use the treated wastewater and not the potable water. But we're constantly making sure that there's an economical basis for the agriculture based on this water. For the boron, I'll let someone else answer.
Well,
I can try to. Sorry.
Just for the boron, one should say that because we make desalination of seawater, so we have to do multi-stage RO, which takes out boron and allows it, because the water after drinking reaches the agriculture. So we do have to make a multi-stage RO when treating seawater.
And just to be more specific, boric acid are across the membrane, the osmosis reverse membrane. And what they are doing in the second stage is increase the pH by NOH, boric acid coming to be borat, which is a bigger anion molecule and is not crossing the membrane. Of course, it costs more because you need another stage, but you can remove almost all the boron. You need to decide what level you want, and by mix different water, you can arrive to the concentration you decide. Our decision is the concentration is 0.4 ppm in drinking water. In the effluent, it's increased a little bit.
Well, thank you. I think we've had a very good discussion. And one thing I'd like to point out to people who are watching this, at least in the United States, is that as Danny talked about making the water available for farmers at a lower cost as because the treatment is paid for by the government, there are externalities that are part of that. In the United States, we have that same issue with freshwater, in that a long time ago, we made the decision that supporting agriculture through water projects was a good thing for the whole country. And so we also have the same things. It's just in different places where we subsidize farmers' agriculture. We subsidize water in the United States to help bring food prices down and to help keep food prices low. I would have to say that as a summary, this has been, or to summarize, this has been a great opportunity for us to come together and talk. And I hope that we can look forward to future collaborations between Israel and the United States to further look at reusing wastewater and reusing lower quality waters to be able to expand the entire water portfolio in the United States. With that, I will thank our speakers and those who have participated in the panel, and I will turn the time over to Omer.
Thank you very much for a great day and great discussion. Thank you, partners, panelists, speakers, and organizers for a fascinating and beneficial day. WaterEdge creates a vital source of water, which means more water for domestic sector, industry, agriculture, public landscapes, for nature, and is essential to cope both with water scarcity and environmental pollution. As Dr. Mark Moorman said 2 hours ago, we must keep building our relationship with water and with water reuse. And the secret, as Dr. Moorman said, the secret sauce is trust in experts and technology. And I would like to add also standards. This relationship is also reflected in monitoring of water and crops, as was demonstrated today, both by the researchers and the tech experts. The discussions in the last 3 days raised the importance for integrative approach to water reuse, and especially the connection between policy, research, that is the base for standards and regulation. And just for the great examples that we have today at the beginning of the day, how to regulate detergents in order to reduce boron and many more, research and development, technologies and infrastructures. Implementing all these elements in one ecosystem is an ongoing challenge. In the last 5 months, dozens of leading experts worked together to identify where our countries share common goals, practices, and interests in the fields of water reuse. We believe that highlighting these various use cases for water reuse could give and provide further ideas on ways to collaborate in several fields. For example, research on water reuse broadly, as well as the use of in agriculture, as was discussed today, data-driven policy, as was emphasized through all the 3 days, standards, regulation, and risk management for water reuse, integrated water resource management and infrastructure that allow efficient continuous supply of water for different needs, potable, agriculture, industry, nature, and others, R&D, and translating research and knowledge into innovative technologies and solutions, and mutual experience in our countries, and the use of mutual experience in our countries create knowledge, test in beta sites, and turn ideas into reality in the field. The ongoing collaboration between the US and Israel generates common language and understanding of the set of standards and considerations for water reuse. This mechanism, together with platforms to foster innovation and collaboration, and such examples were discussed today and yesterday, could make a global impact. We look forward to keep developing the collaboration with EPA, Water Reuse Association, FDA, and USDA in the framework of the Water Reuse Action Plan. Next slide, please. Last slide. Please feel free to reach out with questions to EPA in the email listed here, and the Israeli partners directly or through the Israeli embassy using the emails here regarding any question or further ideas on water reuse and water reuse collaboration. Thank you, everybody, speakers, partners, organizers, all people that support, and of course the audience, for this great conference. Thank you very much, and hope to be in touch soon.
Thank you very much.
Thank you.
Thank you.
Thank you.
Thank you very much. Pleasure being with you.
Thank you.
Have a nice day.